Hello everyone! For today's video is all about the reproductive cyclicity. The follicular phase consists of four major events. They are 1. Elevated gonadotropin secretion from the anterior lobe of the pituitary to follicular growth and preparation for ovulation. 3. Sexual receptivity and for ovulation. The driving force for the initiation of the follicular phase is luteolysis. Luteolysis causes the corpus luteum to become non-functional. The negative feedback by progesterone on the hypothalamus is removed, and GnRH is released at higher amplitudes and frequencies than during the preceding luteal phase. Primary steps leading to preovulatory LH surge. The hypothalamus plays an obligatory role in regulating estrous cycles because it produces gonadotropin-releasing hormone GnRM that is responsible for stimulating the release of the gonadotropins FSH and LH. The tonic and surge centers in the hypothalamus control GnRH release. The surge center responds dramatically to high blood concentrations of estradiol. The profile of tonic release is characterized by having many small pulses or episodes. These pulses have various frequencies and amplitudes depending on the degree of neural activity rate of firing in the tonic center. Thus, as with many neurally controlled hormonal profiles, this pattern is referred to as an episodic profile, surge center, and another hypothalamic center that is responsible for the preovulatory release of GnRH that stimulates a surge of LH, causing ovulation. Anatomically, the surge center consists of 3 nuclei called the preoptic nucleus, the anterior hypothalamic area, and the suprachiasmatic nucleus. The release of GnRH by the tonic and preovulatory centers in the hypothalamus may be compared to water faucets. Tonic basal secretion is analogous to a leaky faucet from which small quantities of water drip from the faucet over a relatively long period of time. GnRH secretion from the tonic center appears to be spontaneous, but is influenced by progesterone. Secretion of GnRH from nerve terminals in the tonic GnRH center occurs in periodic pulses. Each GnRH pulse occurs because of simultaneous depolarizations of several GnRH neurons. Each GnRH neuron secretes a tiny quantity of GnRH, and summation of these small quantities causes a pulse or an episode to occur. Follicular dynamics is controlled by FSH and LH and involves both growth and death of these follicles. Though the follicular phase comprises only about 20% of the estrous cycle, the process of follicular growth and degeneration known as follicular dynamics occurs continuously throughout the entire estrous cycle. Dynamics of antral follicles. It consists of recruitment, emergence, selection, dominance, and atresia. The dynamics antral follicles involve processes. These processes are recruitment or emergence, selection, dominance, and atresia. Atresia, a degeneration. Recruitment or emergence, the phase of follicular development in which a cohort group of small antral follicles begin to grow, emerge, and secrete estradiol. The process of atresia involves far more follicles than does the process of dominance. In fact, over 90% of ovarian follicles undergo an irreversible degenerative process called atresia. The word atresia in the follicular context refers to the closure or disappearance of the antrum that accompanies the degenerative changes. Mid-cycle estradiol increases and declines with each follicular wave, but blood concentrations are low. After luteolysis corpus luteum regression, follicles of either the second or third wave develop into a dominant preovulatory follicle. This ovary contains many small antral follicles, SF. More follicles appear in the ultrasonographic image than appear in the photograph because ultrasound imaging allows observation of follicles beneath the surface of the ovary. Fluid-filled cavities generate a black image, while dense tissue like the ovarian stroma generates a gray to white image. This ovary contains 3 medium antral follicles, MF, and a corpus luteum. See all that appear in both images. The follicular phase has been designated as the initial recruitment phase, involving a continuous recruitment of primordial follicles into a growing follicle pool that terminates with atresia. The second has been termed cyclic recruitment. Cyclic recruitment starts after puberty and is the result of elevated FSH levels that occurs during each cycle. In the face of rising progesterone concentrations during metastress, small quantities of FSH are secreted from an anterior pituitary. This small amount stimulates the emergence recruitment of small antral follicles. These small follicles begin to grow and secrete estrogen. The primary role of LH is to promote final growth and maturation of dominant follicles and to stimulate ovulation. During metastress and distress, the LH pulse frequency is maintained at about 6 pulses per day or 1 pulse every 4 hours. This pulse frequency does not provide enough LH to stimulate waves, and all of these follicles undergo atresia. FSH and LH are regulated differently. FSH secretion is controlled by estrogen and inhibit that is secreted growing follicles that exert negative feedback on the secretion of FSH by the anterior pituitary. Emerging or recruited follicles and early selected follicles are predominantly FSH dependent. Larger selected follicles and dominant follicles are predominantly LH dependent. Follicles with high numbers of LH receptors become preovulatory follicles. During follicular development, LH binds to LH-specific membrane receptors located on the cells of the thecae interna of the developing follicle. Testosterone then diffuses out of the cells of the thecae interna and enters the granulosal cells. The granulosal cells contain receptors for FSH. Cells from rodents in estrus are cornified like that of skin. Cornified cells are irregular in shape and appear crusty using the microscope. In other species like the dog and cat, sheets of squamous cells indicate estrus. Changes in vaginal cytology are species unique, and their appearance has various clinical interpretations. For estradiol synthesis, the cells of the thecae interna contain receptors for LH. Thecal cells produce testosterone that diffuses into the granulosal cells that contain FSH receptors. Binding of FSH to the granulosal cell receptors causes the synthesis of enzymes that are responsible for the conversion of testosterone to estradiol. Elevated estradiol coupled with low progesterone induces profound behavioral changes in the female. During the follicular phase, the female becomes sexually receptive and copulation takes place. Hyperemia, local elevated blood flow, is thought to be controlled at the tissue level by histamine and prostaglandin E2PGE2. In addition, there is elevated local blood flow to dominant follicles. The cells of the thecae interna begin to produce progesterone instead of testosterone. This local elevation of progesterone is essential for ovulation because progesterone stimulates synthesis of an enzyme called collagenase by the thecae interna cells. Prostaglandin E2 is helped the follicle remodel itself into a corpus luteum. After ovulation, the follicle receives its direction for this reorganization from prostaglandin E2, thought to activate a substrate called plasminogen. Among mammals, there are 2 types of ovulators. These are known as spontaneous ovulators and reflex-induced ovulators. The reflex-induced ovulator requires stimulation of the vagina and or cervix for ovulation to occur. Camelids appear to be modified induced ovulators due to the presence of seminal plasma in the female reproductive tract appears to be more important for inducing ovulation than tactile stimulation like in felids. Folliculogenesis and ovulation can be induced artificially using various hormones that has two main approaches, which are the hormonally induced ovulation generally coupled with induced estrus and superovulation. Hormonally induced ovulation requires premature luteolysis, while superovulation is due to an abnormally high number of follicles that are selected followed by ovulation. The picture on the left screen is the hyperstimulated camel ovary that was exteriorized through an incision in the lumbar fossa. The camel was in estrus. There are 13 follicles approaching ovulation. The picture on the top right is the cous ovary that was hyperstimulated with gonadotropins. Ovariectomy was performed on the day of estrus. And last is the picture on the bottom left is superovulated cow that ovary has 9 corpora hemorrhagica, all numbered, indicating individual ovulation sites. Ovariectomy was performed 3 days after estrus. During this period, when oocyte growth involves formation of a large cytoplasm and the zona pellucida, some of the primary oocytes begin to accumulate larger volumes of cytoplasm and develop a translucent band around the cytoplasm known as the zona pellucida that is formed during the secondary follicle stage. An important development during this stage of maturation is the establishment of junctional complexes between neighboring follicular cells and the oocyte. For the final maturation and resumption of meiosis occur near the time of ovulation. Once the follicle has entered the dominance phase, the oocyte becomes poised to resume meiosis. It is believed that when the oocyte reaches a critical minimum size, it gains the ability to resume meiosis when the ovulatory LH surge occurs.